Dr. Julia Burnier, who is an assistant professor in the Department of Oncology and Pathology. She's also a junior scientist at the MUHC since 2018. So Dr. Burnier completed her PhD in molecular and cell biology at McGill, in which she was focusing on the molecular mechanisms underlying invasion and metastasis. She then went on to pursue a postdoc training. It was at McGill and at the Center of Genomic Regulation in Barcelona, Spain. So this was followed by further studies in cancer genomics, in which she was conducting clinical trials at the Princess Margaret Hospital in Toronto. And her lab aims to uncover the role and mechanisms of tumor-derived molecules in tumor progression and metastasis. So her talk is entitled Circulating Tumor-Derived Molecules in Liquid Biopsy, Biomarkers and Therapeutic Targets. Dr. Burnier, please.
Well, thanks very much for the nice introduction and for inviting me to speak with you. I'm going to be talking about circulating biomarkers and how we can really turn to a novel approach to sampling tumor tissue as a really rich resource to identify new biomarkers and targets. So in terms of my presentation, I'm going to give a bit of an introduction to liquid biopsy. I know not everybody listening is very familiar with this exciting new field. At least in my opinion, it's exciting. And if I've done my job correctly, it will be exciting to you by the end of my talk. And then I'm going to talk sort of separately on two different themes: how we use liquid biopsy in my lab for biomarker discovery to monitor patients, and this is mainly through clinical studies. And in theme 2 of my lab, how we look at what circulates in our body during cancer as a way to try to understand the process of cancer progression and metastasis and identify new ways to stop that process as therapeutic targets. So before I go any further, I'd like to acknowledge all these brilliant people that you see at the bottom of this screen who are members of my laboratory, past and present, and who really are absolutely 100% responsible for all the work that I will be showing today. So before I get into liquid biopsy, I want to highlight why I think liquid biopsy represents an important new step in cancer research. Cancer is marked by extreme heterogeneity, meaning that cancer cells within a tumor are not identical to one another. And we know that even within the same tumor, there are clusters of cells that have completely different molecular and genomic signatures as other cell clusters. You can amplify that heterogeneity even further by looking at a patient that has more than one tumor, or a patient that has micrometastatic disease that is forming in different organs. And then on top of that, we have another layer of complexity, which is temporal heterogeneity. Cancer cells change, they evolve, they change their characteristics to evade the immune system. As we apply different treatments, they modify both their genotype and phenotype to try to survive. So we know that tumors change over time, acquiring new characteristics, and to study the molecular profile of cancer, we rely really heavily on tissue. So what happens now is that a patient might be diagnosed with cancer based on a biopsy, which is a very small piece of tissue, right? Or they might undergo surgery where we have access to a bit more tissue. And a lot of the decisions that we make in terms of best treatment, in terms of prognostication, of identifying the particular molecular subclass that tumor falls into, is based on that initial tumor tissue. But we can't do a surgical biopsy or a surgical resection over and over again, right? This is very invasive. It's not possible. So we need a better non-invasive way to monitor and study disease that represents this heterogeneity, where we're not looking at just one small piece of a tumor, but that we're looking at the tumor holistically as a whole, and that we can sample over time. And I think liquid biopsy addresses this need very well. In a very simplistic way, for those who don't know what a liquid biopsy is, it's sampling disease using a biofluid. And that biofluid can be blood, which is the most common type of liquid biopsy, but it can be other ones. And I'm going to talk about this in my talk. It can be saliva or urine. And basically the idea is to use these biofluids to sample disease. And the most studied type of liquid biopsy is in the context of cancer. And as a cancer biologist, this is what my research program does. Within these biofluids, we can identify circulating tumor cells, right? Cancer cells that have left the primary site and are circulating. And these cells are very likely the ones that are responsible for metastatic disease. We can identify nucleic acids like DNA, which we call circulating tumor DNA. And I'm going to talk about this as a biomarker. And we can also look at extracellular vesicles. These are small vesicles that are released by cancer cells. And you can think of it as a sort of mini representation of that cancer cell. And we know they play a big role in cell-to-cell communication, and particularly in metastasis. So all of this gives us a really rich source of biomarkers and therapeutic targets. And for a long time, this would not have been possible because we didn't have the techniques that were sensitive enough to pick up these molecules. If you imagine what's in your blood, all the different cells, normal cells, disease cells, all the different molecules, all the different nucleic acids, proteins, it wasn't possible to identify these entities specifically. But thanks to so many advances, our sensitivity has grown. And now we're in an era where we can look at these things. So I think it's a really exciting time for cancer research. So what are some of the advantages of this? It's minimally invasive, right? If we think about a saliva swab or a blood sample, it's quick, it's much less costly than a surgical biopsy. And really importantly, it can be repeated. So we can do this over time as a way to monitor patients, as a way to say in real time, what is happening in that patient's disease? How have the cancer cells changed? What new signatures can give us insight into how best we can treat that patient? And it represents the whole disease, not just one tumor, but it represents evidence of if a patient already has some micrometastases that have formed, if they have more than one tumor. So I think it's a very comprehensive way to look at cancer. So like I mentioned, I'm going to talk into two themes. The first one is on circulating tumor DNA as a clinical biomarker. So I want to introduce this topic. For those of you who don't know, circulating DNA is released by all of the cells in our body. In general, what we call cell-free DNA, which is the DNA that is released, originates mostly from cellular turnover. So that can be apoptosis, necrosis, any type of cellular stress will cause increases in cell-free DNA secretion. And also there's evidence of active secretion as well from cells. But we need to be able to distinguish cell-free DNA that comes from all of the cells in our body. And if you imagine, for example, in a blood sample, you'll have cell-free DNA that comes from our hematopoietic cells, from our endothelial cells, from our fibroblasts. So how do we identify the cell-free DNA that comes from tumor cells, which is what we call circulating tumor DNA or ctDNA? Well, we can look at things that are inherent to the cancer cells. The most common one that we can look at are somatic mutations. Is there a mutation that has triggered this cancer that we can then use to find the sequences that have come from the cancer cells themselves? But there are other ways as well. Methylation profiling is very big right now, copy number alterations. And I'm also going to talk about viral sequences in cancers that are driven by viruses. And in the last 10 years or so, many groups have looked at the frequency of cases with detectable circulating tumor DNA. So on the left, you see a colorful graph that shows the number of patients that have positive ctDNA in different cancer types. Okay, and this was a study that was done, a pan-cancer study, and you can see that in bladder and colorectal and esophageal, ovarian cancers, these cancers have a really high frequency of patients that have detectable circulating tumor DNA. As we go along towards the pink color, you can see that gliomas actually have quite few number of patients with positive circulating tumor DNA, and this is quite interesting because the field has progressed. Brain cancers, in a way, are a little bit, this was done in blood, and brain cancers are a bit protected from the blood based on the blood-brain barrier. So in recent years, people have looked towards cerebral spinal fluid as a source of circulating tumor DNA from patients with brain cancers, and it's been found to be a really powerful source of biomarkers. So all to say is that the choice of biofluid, of which liquid biopsy to use, has to be thought about, right? What is most anatomically correct? And then on the right, you can see that circulating tumor DNA has certainly correlated with disease burden. So while we do see ctDNA in many primary cancer patients, in the metastatic setting, most patients have circulating tumor DNA presence of circulating tumor DNA. And so what are the applications? We can use it for early detection of both primary and recurrent disease, right? If it's present. Really importantly, and what we do in my lab, is longitudinally sampling patients so that we can monitor for things like treatment response, to see if a patient has relapsed or is developing metastasis, and if we can do that prior to a patient having symptoms or having radiological evidence of disease. But ultimately, the goal is to bring this to the clinic where we can make treatment decisions for patients based on the molecular changes that are happening that we can identify in these liquid biopsies. And that's really the goal of precision oncology. Oops. In recent years, the number of publications that has grown in the circulating tumor DNA field is monumental. People are looking at circulating tumor DNA in studying resistance, to track tumor burden, looking at minimal residual disease in many different cancer types. And ultimately, it's become very clear that it's the future as a cancer biomarker. One pivotal step, I think, was the FDA approval of the first liquid biopsy test, which came in 2016, which was the EGFR mutational test, which meant that patients could be tested for non-small cell lung cancer patients could be tested for an EGFR mutation either in tissue or in blood, and clinical decision could be made to put that patient on an anti-EGFR or an EGFR inhibitor based on either the tumor tissue or the liquid biopsy. So it was really the first validation that clinically this could be used as an assay. And since then, luckily, we've had many others. So what do we do in my lab? We look at circulating tumor DNA as a clinical biomarker. We isolate cell-free DNA from patient samples, and I'm going to talk about which samples we use. We do both somatic mutational screening, or we look at viral DNA detection. And so for somatic mutational screening, we could look at driver mutations, things like KRAS or BRAF, depending on the tumor type. We can look at mutations that are associated with resistance, if our question is to try to monitor a resistance to a particular therapy. But there are some cancers where we don't have obvious mutations for us to track. And so we can turn to viral DNA sequences for example, cervical or head and neck cancer, which are driven by HPV virus. In terms of techniques, there's many different techniques that can be used. For those of you who work in any type of mutational testing, you know, we can use different forms of sequencing, and we can also look at most commonly digital PCR. Sequencing in the last few years has really gained traction in being able to give us really high throughput information about liquid biopsy samples. However, we have to remember that we're looking for a very small proportion of circulating tumor DNA in a background of a lot of cell-free DNA. So we want to maximize sensitivity. So in my lab, we use a lot of digital droplet PCR because we can get to a sensitivity of about 0.01. What does that translate to? That means one mutant copy in a background of 10,000 wild type copies, right? So we're really increasing the sensitivity of our assays so that we can pick up these very rare fragments. So we have a blood sample. We have a way to distinguish that normal from cancer DNA. As I've told you, somatic mutations, viral sequences. And then in my lab, we do a series of 3 main steps. One, we use synthetic DNA, G-blocks, as I'm sure many of you have used, to verify that our assay works. Then, when possible, we turn to a cell culture system. Is there a cell line that has the mutation that we're looking for or the viral sequence that we're looking for? Can we quantify the cell-free DNA from that culture system to verify our assay? And only then do we move to clinical samples. And this sounds like a complex process, but we can actually do this very quickly. And so in my lab, we've looked at liquid biopsies from many different cancers, and this is actually a bit outdated. We have cervical cancer there as well now. And we can look at blood, which is the most common source of ctDNA, but also urine, saliva, and many other analytes that can be anatomically appropriate for us to look at, and sometimes even less invasive. For example, a saliva sample. So in including so many patients now for ctDNA testing, what it allowed us to do is to create sort of a bank of these assays, of these validated assays using our 3-step process, so that if a new patient needs to be monitored with circulating tumor DNA, we're not starting all over again. And this is a screenshot, but we have many, many more now mutations represented in our bank, which tends to encompass a large proportion of the most common cancer driver events. So we can really, if there's a patient with a pancreatic cancer patient, we have our KRAS mutations there, and we can go right to testing the patient's sample. So I think it's a really real-time assessment of a patient's disease. So what kind of results do we get? This is a typical case. This patient was a gentleman with metastatic colorectal cancer who had 4 longitudinal samples during the time that he was on our study. He had a sample taken at diagnosis of his metastatic colorectal carcinoma. He underwent surgery, a surgical resection, and for which we had 2 samples. And then this patient had a recurrence and unfortunately died from his disease. And you can see on the left that the circulating tumor DNA levels really recapitulated that disease landscape. We had high levels at diagnosis that decreased with effective surgical treatment and can be seen to increase at recurrence. And this is compared to, for example, CEA, which is a good biomarker in this context, that also is effective, but, for example, would not have detected the diagnostic time point for this particular patient. What I want to go into is really talk about one particular study to give you a sense of how we can use these circulating molecules as biomarkers. So I'm going to talk about head and neck cancer, and this is largely the work of a graduate student in my lab named Ty Ferrier. And the incidence of head and neck cancer overall has been decreasing, but HPV positive head and neck cancer is increasing. And what's interesting is that this is a disease that head and neck cancer is historically associated with more prevalence in men, heavy drinkers, heavy smokers, later in life. However, HPV positive head and neck cancer is associated with younger patient cohorts. So these people are relatively young. They are not the heavy drinkers, heavy smokers. And while their prognosis is generally okay, the treatment can be very devastating to these patients. They have usually a combination of surgery and radiation therapy. It leaves them with disfigurement in the facial region, difficulty producing saliva, difficulty swallowing, speaking. So these are major quality of life issues in a relatively young population that still has many years, hopefully, to live. So a lot of the literature and a lot of the field has been trying to identify, because these patients have generally a good prognosis, is there a way for us to de-escalate treatment in certain cases to be able to say with more certainty if a patient has a better chance of not progressing, that maybe that patient could be spared from some of these very difficult treatments. So we've teamed up with Dr. Sadeghi and Zaytouni to look at head and neck cancer using HPV sequences. And we've recently added cervical cancer as well, but I'm going to be focusing on the head and neck cancer cohort. With these patients, we do take blood samples, but we also take a saliva sample, which is a very non-invasive, right? Sampling methodology, and we've been testing both analytes concurrently. We test these patients before they receive surgery, so at diagnosis, and then they're treated. We take another sample, and then we monitor these patients over time. And we've included 70 patients so far. And what was very clear to us is that there is a very significant difference between circulating tumor DNA levels before and after treatment in both plasma and saliva, meaning the ctDNA levels decrease substantially after treatment. While virtually all of our patients are positive for ctDNA at diagnosis, most of them go down to either 0 or very low levels following curative intent surgery. And you can see that plasma and saliva here behaved very similarly, but there are a few differences. And this is something that we're still investigating. So you can see in A that the number of patients that are positive pretreatment is almost all of them compared to very few of them post-treatment. What's interesting is that if you look at the bottom graphs, when we correlate the presence of circulating tumor DNA with evidence of tumor, okay, post-treatment, okay, so this means maybe a patient had a surgical resection, but there were positive margins, or they were still able to find disease post-treatment. We see a very strong correlation between presence of ctDNA and what we call minimal residual disease, right? And minimal residual disease, or evidence of tumor, correlates with outcome, meaning we have a really quantitative way to say if a patient has an increased risk of progressing later. And maybe these are the patients that would most benefit from continued treatment. And this is something that we're looking at. While the patients that go down to 0 after a very successful surgery, perhaps those patients could be spared from some of the subsequent treatment that they generally receive. What I haven't shown is that while circulating tumor DNA has really correlated with disease progression with treatment response, we see huge differences between patients in level of ctDNA. What's become clear to our group is that we can compare patient X to him or herself by comparing pre and post samples, by looking longitudinally over time, by doing the types of graphs that I showed you with the colorectal liver metastasis patient. But if you compare between patients, it's very difficult because these patients have very different levels of ctDNA. And in recent years, people have started to look at that. What causes cell-free DNA to be emitted? Even things like exercise can cause surges in cell-free DNA. So how can we better understand circulating tumor DNA emission mechanisms and kinetics so that we can interpret our liquid biopsy data a bit better, so that we can identify how to quantify those levels? So my lab turned to using culture models to quantify cells, the release of cell-free DNA from cancer cells. So we put cancer cells in dishes with culture media, and then we subject them to all sorts of different treatments. We subject them to chemotherapeutic agents to radiation. We also induce cytotoxicity that cells undergo with heat, with specific agents that will make them undergo apoptosis, necrosis, cellular senescence, cell cycle arrest, to try to understand what causes cell-free DNA emission. And what was very clear to us from the beginning is that as soon as cells undergo any sort of cellular stress, whether it's a treatment, whether it's a specific agent, they will secrete cell-free DNA in a dose-dependent manner. And when we look at, for example, apoptotic markers or necrotic markers, using, for example, an XN5 or PI, we can correlate what's happening to these cells, what cellular process they're undergoing, whether it's apoptosis or necrosis or cellular senescence, to the emission of cell-free DNA. And in fact, apoptosis, which is one of the mechanisms of cell death that many chemotherapeutic agents act through, is the biggest contributor to cell-free DNA emission. So it's something that we have to think about as we think of when to sample patients. For example, during treatment, should we wait until patients have undergone all of their chemotherapeutic treatment before taking a sample, so that we don't think they're still positive only because they're in that active secretion phase of cell-free DNA? What's also really interesting to us as a lab that focuses on metastatic disease is that we saw very clear, very clear decreases in cell-free DNA emission in cells that are undergoing senescence. So if we think about metastasis, what happens? There's a primary tumor, those cells circulate in our body. Even if the patient is really well treated and they have no evidence of disease, 2, 5, 10 years later, they develop metastasis. And one of the prevailing theories on this is that the circulating tumor cells can undergo dormancy or senescence, where they lay dormant in different organs of metastatic organs, such as the liver or the lungs or the bone marrow, where they're awaiting some microenvironmental cues that will awaken them from that dormancy. So it was really interesting for us to find such a clear correlation between senescence and decreased emission of cell-free DNA, meaning we really can use this as a biomarker to monitor these cells and their release of cell-free DNA, hopefully picking up when those cells awaken into metastatic disease. So this is a really, I think, exciting part of the liquid biopsy world, where we turn back to basic biology to understand clinical liquid biopsy data. And in fact, we've even seen that we can use this as an application for preclinical drug testing. Many people here listening today might look at testing some new therapies, and you can use cancer cells, you can use organoids, PDXs, and to be able to assess treatment response, it's not just cytotoxicity, it's not just cell death or tumor burden. Do we have good biomarkers to tell us what's happening? And so we've been doing some preclinical testing for specific drugs. This is just one example. This is mifepristone. That was being investigated. It's being investigated in a number of different cancer types, including ovarian cancer, including melanomas. And so what we tried to see is, could we test ctDNA as a preclinical biomarker for mifepristone, for the effects of mifepristone? And so you can see that, so mifepristone causes cytotoxicity in these cells in a dose-dependent way. And in fact, we could recapitulate that dose-dependent dependency using cell-free DNA just in the culture media. So I think this is a really fast and a really cheap and available way to monitor drug response and even resistance in preclinical testing. So hopefully I have convinced you that I think ctDNA is a really powerful clinical biomarker. What's important is that it gives us information in real time. We're not waiting huge amounts of time, and we're not reliant on banked biopsy or banked surgical tissue that has been sitting somewhere for 3 years. This is what the patient's disease looks like today. And we can do this in a non-invasive way. But I also think there's value to keep looking at in vitro models of cell-free DNA emission, because it really helps us to understand the kinetics. It helps us to better interpret the clinical data, because up to now, we really haven't, I think, interpreted it in the best way. So the second part of my talk is on a different liquid biopsy analyte, which are extracellular vesicles. And like I mentioned, these are small membrane bilayer vesicles that are secreted by virtually all the cells in our body. And even if we stay away from the concept of cancer, extracellular vesicles for a long time were thought to just be garbage that the cell releases. And in fact, we now know that they're much more than that. You might have heard terms like exosomes, extracellular vesicles, microvesicles. These vesicles are now known to be key mediators in cell-to-cell communication. So how does that happen, right? Well, EVs are secreted by all cells in our body, and then I'm going to focus on cancer, and they're internalized by recipient cells. And in those recipient cells, they dump their cargo, which could be proteins, nucleic acids, and exert a number of different effects. Okay, so they act in the context of metastasis. They can even act as long distance messengers. And they seem to be particularly important in communicating with the cells of the microenvironment to prime that tissue for colonization by circulating tumor cells. So for some of you that might not be in the field of cancer metastasis, this is the pre-metastatic niche formation idea. The idea that cancer cells are released from, let's say, a primary breast cancer, and they're circulating in our bodies. They might become dormant for a while, but they're sitting in those metastatic spaces, in the liver sinusoids, in the lungs, in the bone. And due to some communication between those cancer cells and the microenvironmental cells, the normal cells of those organs, that organ begins to change. We see increased vascular permeability. We see extracellular matrix remodeling, recruitment of immune cells, angiogenesis, immune modulation as well. And that's when cancer cells can then thrive and form metastasis. And the EVs have really been implicated in this process as acting as mediators, as the messengers from the cancer cells to the recipient cells. So a few years ago, you can see here at the bottom, Tupten Sering in my lab, wanted to ask exactly this question. What happens when tumor cell-derived extracellular vesicles are given to cells representing the liver metastatic environment? So we took cells representing the liver parenchyma, so hepatocytes and fibroblasts, and we took EVs from primary and metastatic cell lines, and we seeded them on these normal cells. The normal cells are here stained in blue, and the extracellular vesicles are tagged in green. And we saw that they're uptaken, that they're internalized by these recipient cells. And he went a little bit further to ask, well, what happens to these cells? Is their behavior the same? Is their phenotype the same? So we did migration and invasion assays on these, again, normal recipient cells. These are fibroblasts, hepatocytes. And these hepatocytes and fibroblasts had significant increases in their migratory capacity and their invasive capacity, meaning they began to change. They were delivered some sort of tumor-promoting content that modified their behavior. And we saw the same effect in vivo. We took microenvironmental cells, again, hepatocytes and fibroblasts, and we exposed them to extracellular vesicles from both normal and malignant cells. And after 3 weeks of exposure, we injected these cells into mice. And it was only the cells that had been exposed to cancer EVs that formed subcutaneous tumors, whereas the cells that had been exposed to normal cell EVs didn't. So what it led us to believe is, okay, EVs are communicating with these recipient cells and they're delivering something. But what? What is it that they're delivering? And how are we going to be able to make sense of which molecules that are being delivered are actually important in the metastatic process? So we ran high throughput proteomics to look at the differences in extracellular vesicles in primary, metastatic cells, normal cells, to try to identify EV protein signatures. Can we find specific proteins that could be implicated? And we found thousands of proteins, as one does in these types of large screens, but we did see enrichment for some key proteins that have been, by other groups, also shown to be enriched in extracellular vesicles from these models. So things like integrins. If anybody here has studied integrins have been highly implicated in site-specific metastasis. These sit on they're heterodimers that sit on the cell surface and on the EV surface, and likely play a role in leading cells and EVs to specific sites and being internalized. So we saw specific integrin signatures. We also saw extracellular matrix proteins like fibronectin. And so we validated these, as you can see on the bottom, co-enrichment analysis with CD63, which is an extracellular vesicle marker. So this was the first indication to us, and in a body of literature that has found similar results, that there are very specific proteins that likely aren't just randomly housed in extracellular vesicles, but enriched in a way and causing some of the effects that we see. But we identified thousands of proteins. How can we now test them one by one? And that was a very daunting task. So we turned to engineering. Can we use a synthetic system to study cancer EVs? So Ruben and Shaima, whose pictures are shown here, are largely responsible for this work. This was a collaboration between my group and an engineering group from ETS, Concordia, and UCAM, where we said, okay, we have these vesicles and they're filled with all sorts of things. And if we start knocking down everything, this is going to be a very tedious task. So it's very difficult to try to identify which proteins or which combinations of proteins might be playing a role. But as clever engineers, they always have a solution. And they said, well, what if we took synthetic vesicles, liposomes? And I knew nothing about liposomes, but I quickly learned a lot about liposomes, that liposomes in many ways resemble natural vesicles. They're lipid bilayer structures. They have a core that can house content, and they can have molecules integrated into their lipid bilayer. So we said, this is quite a nice model. So Ruben in the lab, in a very clever way, coined the term, could we progressively mimic our cancer EVs? We've characterized cancer EVs now from many different cancer types using primary, metastatic, normal cell EVs. We've looked at their size, their charge, what they have. We've done proteomic analyses. Can we now start to mimic them progressively? Can we take empty liposomes? Can we make them the same size and charge as extracellular vesicles? Can we mimic their outer layer, their lipid structure? And then can we start to integrate them with those proteins that we think could be playing a role? Meaning we now have a way to go one by one, adding our proteins and then adding them in combinations. So we have our EV characterizations. We can now engineer these EV-like liposomes using microfluidics. And we can assess how these EV-like liposomes are uptaken by those same recipient cells that we've done all of our true EV work on. And finally, we can even look in the future to see these liposomes as delivery vehicles to specific cells. And we facilitated this in the lab by doing 3D printed microfluidics. What we had to do in the beginning, we set up a microfluidic system with the pumps, and you have, the organic solvent on one end, you have the aqueous solvent in the other end, and we were making liposomes. But each time we needed to make a different size liposome or change our formulations, we had to make a new chip. So we bought a 3D printer that could make these microfluidic chips very easily, that we could amend our protocol, that we could change the parameters to make different formulations. And so what you can see here, the bottom middle panel is a very homogeneous production of these synthetic liposomes. And in fact, this is largely the work of Shaima. We were able to create synthetic EVs, EV-like liposomes, with the same size and charge as our cancer EVs. So you can see the targeted and obtained parts of this table, where we had the targeted diameter or targeted zeta potential, which is the charge of these EVs. And you can see that we came very close to approximating those parameters in our synthetic model. But does it work? Does it work in terms of uptake? Can recipient cells internalize these synthetic EVs in the same way that we were showing uptake in our cancer-derived EVs? So here on the left is the same figure that I showed earlier in the talk, where we see cancer EVs being uptaken by fibroblasts and hepatocytes. And here on the right is our synthetic model. I apologize that the colors are reversed. So in the right, it's in red that you're looking at, but you can see the uptake in red of these synthetic liposomes in a very similar time frame, 12 to 24 hours, that we were seeing in our natural EVs. What is clear from this figure is that when you modulate size and charge, you see differences in uptake, and we wanted to look a little bit closer at that. So what we built was a library of different synthetic formulations, which we could test for uptake, different sizes, different charges, different now lipid profiles. And we wanted to see how this impacts, each of these different parameters impacts internalization. So I hope this video will work. But you'll see that over time, this is one particular formulation, is being uptaken over 24 hours on these hepatocytes. And you'll see in red signal that they're being internalized into the cytosol of these cells. So this was really exciting to us because we've developed a synthetic model, which we can then load with the proteins that we're interested in studying. And we've done this in vivo too. We've said, do these liposomes, will they reach target organs? And we have very preliminary data. We're only at the beginning of this, to show that these nanoparticles can make it to where we expect, which are the liver and the kidneys. So as I mentioned, it was clear to us that as we modulate some of these parameters, particularly size and charge, which are very important in that initial uptake by recipient cells, we see differences in uptake. So you can see that with increasing size, for example, at the bottom, the liposomes are less uptaken, which makes sense. And so the final piece of this story is, can we load these EVs with protein content, which seems like an easy idea, but it actually was much more challenging than we anticipated. But can we, in one step, make liposomes that have the size, the charge, the lipid profile that we want, that contains a lipid bilayer, and still include protein within its core? And so after many, many tries, you can see at the top right, this is a transmission electron microscopy image of a clear lipid bilayer with a protein core, right? Meaning that we were successfully able to deliver protein. And we did this in two ways. One is we wanted to ensure that we could deliver functional protein. So we use GFP. Can we take GFP protein and include it in these liposomes? And as you can see here, yes, we can. And secondly, can we take the dump from cells, from extracellular vesicles, the proteins? Can we extract the proteins from those cells and from those extracellular vesicles and include them in our liposomes? And we've recently shown that we can. Meaning that we've created these EV-like liposomes with not only the physicochemical properties of EVs, but also as a vehicle to deliver the proteins of interest. So what questions can we start to address with this model? Well, the same question that we had from the beginning: what cargo to load? Do we start with our integrins? Is it a combination of proteins that are required to recapitulate pre-metastatic niche formation or metastatic process? Which microenvironmental cells are important as recipient cells? Which cells of the liver? Do immune cells take up these cancer EVs? Do other cancer cells take up these cancer EVs? And in fact, these EVs also work on cancer cell to cancer cell communication. And then finally, can we redirect EVs by modulating their characteristics in a synthetic model, right? Which has a therapeutic application. If we know that cancer EVs will go always, colorectal cancer EVs will always go to the liver and are responsible for liver metastasis, can we redirect them? Can we stop this process altogether? So in fact, it has big implications for a delivery system for therapeutics. So to conclude, I think we have to start thinking about systemic disease as an early event, meaning that our methods to study and target the disease should also be focused on the systemic nature of cancer, and that includes disseminated molecules. Liquid biopsy represents a really powerful source of that, and I think we're going to see that being used more and more. EVs transfer important traits to recipient cells. We know that. But how they do that, what those traits are, what molecules are involved in this process is very unknown. And I think there's a lot to be done in this field. Synthetic biology and bioengineering can help us. It can facilitate all the challenges that we have in extracellular vesicle research. For anybody in the audience who has worked on EVs or exosomes, it's a challenging field. It's cumbersome, where you just to get a small amount of EVs, you have to culture liters of media. Can we turn to our bioengineering colleagues to help facilitate this study? So with that, I am done, and I'd like to re-acknowledge all the wonderful people in my lab and our sources of funding and collaborators, and hopefully some of you are still awake and we'll have some questions to ask. Thanks.
Thank you very much, Dr. Burnier, for that really exciting presentation. I remember years ago when I was a graduate student, we were working on, we used to call them matrix vesicles. And it was impossible for my boss to get grants because there were a lot of pushback. They used to say that these are artifacts, but it's nice to see how they have come a long way. Any questions, Dr. Jean Trisha?
Hi, Julia. Very, very fascinating work. And maybe this question is just an overture because I'll come over to your office and continue the conversation one day. But yeah. Listen, these EVs are fascinating. We've been doing some stuff with Paraskevas and I in transplant, where we took our perfusates from kidney transplants, and they elute a lot of EVs. And we found that certain EVs with certain mRNAs, they have a propensity to induce perfusion injury in our kidneys. They're predominantly taken up by macrophages, monocytes, and B cells. So my question to you, because I'm not sure, maybe cancer EVs are a little different, I suppose. It's possible, but maybe this is a phenomenon that works all the time. And they're taken up by some of the immune cells of the liver. The liver is full of Kupffer cells and all kinds of other immune cells, and stellate cells, which act like fibroblasts. And so, the liver is like the organ where metastases grow. And so my question to you, because we know these in EVs from perfusates induce preferentially Th17 cells, and they downregulate Tregs. So could the same thing be happening where these EVs prepare the niches for cancer by inducing some form of IL-17, which recruits neutrophils, which then promote pro-carcinogenic in metastatic milieu. I don't know. Does that sound like logical, or I'm not sure?
So my short answer to that question is yes. My longer answer is no. I think you're absolutely right. I mean, we haven't studied the immune cells of the liver as the recipient cells yet. It was part of my CIHR grant that went out in September. So cool. I'll know January 18th if we're going to look at that. But okay. No, but I think this is a huge field, and not just in cancer, like you mentioned, in transplant, in transplant and injury, in inflammation. We have to look at these EVs not as I'm studying them in the context of cancer because that's what I do, but actually these are communicators, right? And I think they're going to really shed light into how that environment is being modulated, and immune cell function is certainly being affected by extracellular vesicle uptake. And so Kupffer cells, that's on our radar to do. I think this is going to be a really exciting new direction, but even all the immune cells. And I would even go further to say, is it only in the metastatic organ? And I think this is mainly where it happens, but even in the blood. What is happening? Are we seeing some modulation in the blood that is causing a systemic change? So I'm totally on board. This is exactly where the field is going.
There was a fascinating paper which I still quote, was published a couple of years ago, maybe 5 years ago now, where they grew non-metastatic breast cancer cells into a soup high in IL-17.
Okay.
And then they inject them back in mice, and they behave like the aggressive metastatic type. It was fascinating. And so I'm wondering if the same thing happens here through the liver and the immune cell upregulation.
Yeah.
I think we used to think as like very dichotomy, regs, bad for cancer, and immune upregulation good for cancer, but I think it's more complicated now.
Yeah, and hopefully EVs will shed some light into maybe a context dependent.
Yeah, that's fascinating. Thank you. Enjoyed it.
Thank you.
Any other questions? So when you collect the blood, you collect in EDT air tubes.
This is for circulating tumor DNA?
Yes, I'm asking because normally when you take blood and you put in this EDTA tubes, you need EDTA to reduce coagulation of the blood.
Mm-hmm.
But then you have the white blood cells which will lyse and release genomic wild-type DNA. How do you? It's
a good question. We've actually tested both. The consensus in the field is to stick to plasma, so EDTA tubes. We actually use specialized tubes called PAXgene tubes because they have a DNA stabilizer. And what's become very clear to us is that the pre-analytical parameters in terms of processing time, all of this is really important in maximizing ctDNA yield. So we use these special tubes that have some sort of DNA stabilizers, which of course the company doesn't divulge, so I can't tell you what's in it, but there's a few companies who make them. We have done them in serum tubes as well. In fact, you can use both. In general, people have been using plasma tubes exactly for the reason that you said, because you have this background that is avoided. What I would say though, because the consensus in the field now is to use plasma, many people who have banked samples and they want to do a liquid biopsy study, and it's all serum because this is the way that we used to do it more, think they can't do it. It's still very, it's still, even if it's not optimal, I think there's still a lot of value in using serum tubes, and the field has flip-flopped a little bit. But in general, we use plasma tubes, and I do see a value in adding these stabilizers to them so that, you know, in the clinic, for example, these tubes can sit for 2 weeks at room temperature and it doesn't impact at all the yield, which is quite remarkable.
Right. Yeah, so I noticed that you are using mostly plasma and not serum, which is yeah. So in a healthy person, you have these infiltrating phagocytes. If you have something, let's say apoptotic or necrotic cellular debris, they'll clear them. So what's happening to the phagocytes in cancer?
Well, I think the DNA can still be cleared, but the point is that the half-life before, I mean, the half-life of cell-free DNA is very short, right? It's likely in the order of minutes, right? So what you're capturing when you do an analysis is that real-time assessment of the cell-free DNA levels. What happens some minutes later is different, but it's in a constant loop of secretion and clearance, secretion and clearance, right. Secretion and clearance.
Okay.
Now, in a cell culture model, we think about this a lot because we don't have those factors. And actually in my lab, we've been toying with the idea of creating a media that more represents blood in terms of nucleases, in terms of those clearance mechanisms, right?
Right.
Whether it's clearance or degradation. But I think in terms of blood, what makes liquid biopsy such a real-time biomarker is that it's in this constant loop of secretion and clearance.
Okay. Any questions? Please just ask.
Hi, Dr. Burnier. Yeah, I have a question. Actually, I'm very fascinated by your EV model that you use to generate the EVs, and it's amazing how you can, I guess, based on the thickness of that like tiny little lithography, you could design, you could approximate, or you could adjust the size of the EVs generated, that's pretty cool. So in your, it's a 2-component system. So you have, I guess, a hydrophobic side, which includes the material for the liposome, as well as and then you have an aqueous side, which includes your soluble proteins. So and you mix them together, that gives your EV, and the soluble proteins, I guess, get entrapped within the liposome. So I guess my question is, like, is there limitations on the types of proteins that you could study? Since you're dealing with like soluble proteins that you're mixing with a hydrophobic component, because membrane proteins, for instance, I guess you'd have to, they're kind of integrated in a natural EV. I guess it'd be integrated directly in the membrane portion of the EV. So is that still a possibility to study in your model?
It's a really great question. And it's something we're working on right now. It's hard for us to know how the proteins will be packaged into these synthetic EVs, right? In an ideal world, we want the transmembrane proteins in the, in transmembrane. We want the cytosolic proteins or the luminal proteins in the lumen. That's how we want. There is a paper that was kind of our inspiration for thinking that this was possible, because I think we were maybe the only ones who thought this was possible, that suggested that the transmembrane proteins do tend to, because of their domains, also integrate into the synthetic EP membrane. The problem is which direction they go into, right? So you might get a 50-50 where some of these proteins are facing the wrong way, right? But that might be okay, because it's not going to be a perfect model. But let's say, unfortunately for us, the first protein we're really interested in are integrins. That's a really hard protein to study for many reasons. One, because it's not luminal. Second, because these are dimers. So we also need the alpha and the beta chains to get right close to one another and to form a dimer for us to actually see any effect. So we were kind of starting with the most difficult scenario. But let's say that half of them are flipped. That's okay. I think what's difficult right now in EV research, and there are so many papers suggesting this protein X is important in metastasis, or protein Y is important in whatever else from EVs. And we have no way to test these individually because we can't knock out everything in an EV, right? So even if it's not a perfect, I can't guarantee we're going to get all of these proteins in the right place. I see them already in the lumen. So, you know, we can get them in there, which was our first challenge. We can get a lipidomic profile on the outside of the EV, the synthetic EVs that resembles the natural occurring ones. That was also something we weren't sure, you know, how successful we would be. So the next stage is to see if we can get these transmembrane proteins or these membrane proteins to sit on the membrane of these EVs. And based on this previous paper, I'm hopeful that we will, but I'll have to update you to tell you if it worked.
Very interesting. Thanks.
Send me positive vibes, please.
Yes, definitely.
Any other questions? So if there are no more questions, thank you very much. That's really wonderful. And I look forward to seeing more in the future. Exciting work.
I really appreciate it. Thank you for having me. And
Yeah.
thank you so
Thanks for
many of you that
presenting.
come out.
Thank you. Bye, everyone.
Bye, bye-bye.
Thank you.